And we're off.
Good afternoon. First of all, I would like to welcome everybody to my presentation. My presentation will focus on some recent findings from the experiment that I conducted in my doctoral research. The title of my talk is Hormone-Induced Defense to Merovingian grammatical infection in rice. We know that plants have various layers of defense against pathogen attack. In some cases, it is pre-existing defense, while in others, it is induced defense that can be mediated by signaling pathways. Induced defense by hormones plays an important role in protecting plants from various biotic and antibiotic stresses. The role of key hormonal pathways in induced plant defense after nematode infection has already been investigated, but the results are often fragmentary and contradictory. Moreover, all documented studies conducted so far have focused on dichotomous plants only. Above all, a study on rice, one of the most important food crops, is yet to be conducted. What are nematodes? Nematodes are multicellular non-segmented animal that can cause diseases to animals and plants. However, our research only focuses on plant parasitic nematodes. Compared to free-living and other parasitic nematode, plant parasitic nematode has a special kind of structure in their mouth called stylet that they use to infect the plants. Among the plant parasitic nematode, Merovingian graminicola is the most important plant nematodes. Losses caused by this nematode can be as much as 80%. They have four different stages in their life cycle. It's called juvenile 2, 3, 4, and adult. The second stage, juvenile, penetrates the root through the root tips. After penetration, they develop by establishing a special feeding site. This process results in special typical hook-like structure called root knot. The adult females lay their eggs inside or outside the root, and juvenile hatch from their eggs and reinfect the same root. Considering the risk effects, our research objective was set to analyze the differential gene expression due to signal compound, that is hormone application and nematode infection, by quantitative reverse transcriptase polymerase chain reaction, and to functionally study the rice plant defense pathways in relation to the application of key hormones against Merovingian graminicola. The following part of my presentation is outlined under the result of 4 experiments. The first experiment will focus on the effect of exogenous hormone treatment on rice seedling growth. Gene regulation analysis in phytohormone-sprayed young rice plant roots was studied in this experiment. The third experiment was on the expression analysis of different defense pathways upon Merovingian graminicola infection in rice root. And finally, we studied the effect of exogenous hormones on induced rice defense against Merovingian graminicola. We used 4 hormones, and these were salicylate, ethylene, jasmonate, and abscisic acid. The concentration of the chemical used in this experiment shown on the right column of the table. In case of infection experiment, 5 days old seedlings were transplanted to specially made PVC tubes filled with sand absorbent polymer. 10 days later, plants were sprayed with hormone. 10 days later, plants were sprayed with hormones, and after 24 hours, they were inoculated with nematodes at the rate of 300 per plant. Data on plant defense parameter, that is, number of galls, were counted when the plants are 30 days age. You can see this is 1 galls, and inside the gall, the red are nematodes. So, I will discuss now some of the results of my experiment. In order to fulfill the research objective, 4 individual hormones were sprayed, and their effect on seedling growth was studied in this experiment. This satisfied the question to resolve the independent role of this hormone. In this experiment, only ABAT treatment had significantly different shoot length and root weight. For other hormones, there were no significant difference in growth parameters. In order to verify the effectiveness of chemical as an inducer of the corresponding pathways in the rice root system due to exogenous foliar hormone application, For each pathway, one hormone responsive gene were selected to see their expression. qPCR analysis revealed that all the hormone responsive genes were significantly upregulated at earlier time points in the rice root system due to responsible for hormone application. At later stages, different genes from different pathways showed inconsistently differential expression. Considering the effectiveness to many endogenous signals like ethylene, jasmonate, salicylate, abscisic acid, hydrogen peroxide, cytokinin, two pathogenesis-related genes, PR1A and PR1B, also selected to check their expression profile to these four hormones. qPCR analysis revealed that both ethylene and methyl jasmonate can effectively regulate both this PR1 gene and PR1B gene at indicated time points of investigation, whereas abscisic acid and BTH was the poor and late inducer of these pathogenesis-related genes. These results clearly demonstrate that ethylene and methyl jasmonate can effectively regulate these pathogenesis-related marker genes in the rice root due to hormone application. Activation or suppression of different defense pathways upon malaria graminicula infection in rice root was studied in this experiment. For each pathway, one defense-related gene along with two PR1 genes were selected in this experiment. Most of the genes tested were significantly downregulated or had returned to non-infected tissue level at indicated time points of investigation. For ABA pathway, both gene transcripts are significantly upregulated at earlier time points upon infection, whereas jasmonate responsive transcription factor B gene upregulates at later stage of infection. Actually, in order to antagonize the rice plant immune responses, Malaria graminicula manipulated the signaling network between the defense pathways for establishing their feeding sites. And the fourth experiment, rice accumulates high level of salicylic acid in young leaves and shoots, and salicylic acid is generally linked to systemic acquired resistance. Three different chemical sources were used to supply the salicylates. Among the three chemicals, only BTH showed some positive significant effect in induced defense for Malaria graminicula. Salicylic deficient transgenic NAMG plants correlated increased but non-significant susceptibility to nematodes. For other three hormones, the induced defense against nematode were investigated in terms of number of galls per plant and number of galls per gram of root. All the three hormones significantly reduced the number of galls per plant. Among them, ethylene and methyl jasmonate are the most effective ones. In terms of number of galls per gram of root, only ABA treatment had no significant difference. So, we can conclude that at earlier time point, all hormone-inducible genes were upregulated due to responsible hormone application. Among the studied hormones, ethylene and methyl jasmonate were the effective inducer of pathogenesis-related marker genes. Most of the genes were significantly downregulated or had returned to non-infected tissue level in nematode-infected plants. And finally, we found that ethophane and methyl jasmonate were the most effective ones in the hormone-mediated rice defense against Malaria graminicula. Thank you so much for your attention. Any questions or remarks?
For practical application purposes, what does this mean? Can we now treat rice through these hormones to?
Yeah, to actually to replace the chemical fungicide that we are using to minimize the disease. So maybe we can apply the hormone
And
to reduce the disease.
How are we going to get the hormone there?
From the company.
From the company, but is it going to be sprayed? Can we induce the hormone into the rice itself? Or?
Yes.
Yeah.
Yes.
So it's through spraying.
Yeah, it's true. It's spray on the root, but it's spray on the plants and foliar spray. But I'm working with root pathogen, and then I try to look the systemic induction of this hormone to the root.
Because it's taken up by the leaves and then it has to go through the entire system down to the roots.
Because I didn't use in the soil because it can be there some effect on the nematodes, the direct effect on nematodes.
I see. Okay. Thank you.
And what were the factors of choice on your hormones? Are they common hormones that are generally used, or you choose your hormones specifically for nematodes or for what?
The hormone that I used here is already have been worked on other pathogen in rice. So there is no reports on the monocot plants. So I'm working on monocot, but there is lots of references and also some work on nematode in dicot. So there is already lots of work in this hormone. This is the most important hormone that involved in different physiological processes of the plants. So these are the most important hormones: sensory, and accessibility. So there are a lot of articles and findings on dicot plants, how they interact each other in other pathogen interaction, and also some reports on dicot and nematode interaction there. But there is no report on monocot and nematode interaction.
That is really interesting.
Yes. And I have already submitted one article. And yesterday I got the news, it's just minor revision, it will be accepted.
Congratulations.
Thank you. I'm really happy because I submitted in plant physiology. Normally they expect the lots of work, but because this is the new work, so they really like the work, and they said, "Oh, okay, we want to publish our journal, but you have to do some, not the experiment, just discussion, right, like that way or that way, like this."
How often do we need to repeat the spraying? Is once enough?
Once 24 hours before inoculation. I did once.
And
That's why you can see in this experiment, it's just to see the activation of the corresponding pathway in the rice root after 24 hours. If you apply in the root, apply in the shoot, what about the pathway? This is the gene for ethylene pathway. It's activated at 24 hours. It's upregulated with gene. And also the jasmonate pathway gene, it also goes up. So it means the pathway activated in the root at 24 hours after spraying.
Suppose that we were to do this in a rice field and we continue to do this, will the nematode populations then decrease because they don't no longer have a feeding ground, if I can call it like that?
Sorry?
Because if you continually do this in a rice field, will that have an impact on the nematode population?
Yeah.
They will decrease, I take it.
Yes.
After some time, is there then a situation where you will no longer need to spray, or do you envisage that spraying will be?
Yeah, it depends on how many nematodes, how much the plants are infected.
Okay, thank you.